So for our final talk, it's my pleasure to introduce to you the recipient of the 2017 G. Evelyn Hutchinson Award, Dr. Philip Boyd, Professor of Marine Biogeochemistry at the University of Tasmania Institute for Marine and Antarctic Studies. So the Hutchinson Award is given annually to a scientist who has made exceptional contributions to limnology or oceanography in the early or middle stages of their career, and who shows promise of continued scientific excellence into the future. And Philip is receiving the award for his pioneering work on the complex interactions of biogeochemistry, multiple drivers of climate change, and their impacts on ocean planktonic ecosystems. He conducts research on a variety of topics and scales, spanning from molecules to ecosystems. And while his research interests are wide-ranging, he is best known for his pioneering work on iron biogeochemistry of open ocean ecosystems and ocean iron fertilization. His large-scale iron enrichment experiments in the Northeast Pacific and Southern Oceans, along with tracer and modeling studies, have demonstrated how iron limitation can control phytoplankton productivity and therefore profoundly influence the carbon and nutrient cycles in the ocean. And his numerous high-profile publications from this work are widely cited, and his 2007 synthesis paper on mesoscale iron fertilization studies, published in Science, stands as a defining analysis of the subject. Philip's work has been central to the discussion of the use of ocean iron fertilization to stimulate CO2 sequestration, which is a controversial method put forward to mitigate climate change. And he's taken leadership roles in educating policymakers on the social and environmental impacts of geoengineering. So Philip is widely recognized to be among the most influential mid-career oceanographers today, and his wide range of interests and versatility ensure continued scientific excellence. So please join me in congratulating Dr. Philip Boyd.
Thank you very much, Kimberly, and apologies to you all because this was supposed to happen several days ago, but I ended up at the ultimate drive-in movie, which was sitting on a plane in the runway in Sydney, watching Hell or High Water with Jeff Bridges for 2 hours while we tried to get the plane sorted out to take off. So anyway, thank you for staying after Bo's talk for this late presentation. And not a very good example to early career researchers to turn up several days late. So anyway, I apologize. And what I'd like to do today is actually refer to a rather different kind of creature. It's one called Hutchinsonia, and this was a paper that was written by one of Evelyn Hutchinson's students who talked about his phylogeny and his biogeography. As you can see, he was an incredibly prolific scientist with 31 PhD students, almost one a year to come through. And the reason I put this up here is really just to use the example of his stewardship and his tremendous legacy to also pay tribute to my postdoctoral mentor and the person who nominated me for this award, Paul J. Harrison, sadly passed away a few months ago. And I don't think it's a stretch of the imagination to say that Paul, too, has got an incredible legacy of academic children and grandchildren out there in the field today. So this is talk dedicated to you, Paul. In terms of thanking my collaborators, and also given that it was late on Friday, rather than give you a role longer than the Lord of the Rings credits, I thought I'd just give you a word cloud here. And as you can see in the swirling in the midst of the cloud, there's a Dave Carl in there, so you know, he's omnipresent. But anyway, a really big thanks to, this is just my collaborators over the last decade. We've had a lot of fun. We've done some, I think, interesting science, but it's always been enjoyable. So thanks to you all. Again, taking the lead from Evelyn Hutchinson. This is an article that was posted by another professor at Yale, David Post, in 2015. And he really points out the sort of prescience of Hutchinson's work. That's why it's such an honour to be here today, because he had that very multifaceted approach to understanding the impacts on ecosystems. And David Post sort of mentions how we can really learn from that multifaceted approach to try and tackle one of the big issues that's facing us at the minute, that of sort of ocean global change biology. And so I'm going to focus on that and try and take a sort of leaf from Hutchinson's book to sort of try and take a holistic view of it for the rest of this presentation. And so this is the title of the talk, and really what we want to do here is to sort of better understand how changing ocean conditions are going to influence a wide range of processes, diversity, biogeography, physiology, microevolution and so forth, and how that will then play out for marine life, and then again the influence on ocean ecosystems, but then also on marine biogeochemistry. So I'm going to sort of step you through that. And these are some of the challenges we face. We have this huge raft of permutations. Essentially what we're doing is rebooting the flashcard of the ocean. We're concurrently altering oxygen, we're altering the nutrient fields because we're changing the degree of communication between the surface and the ocean's interior. We're also going to be, with loading up the ocean with anthropogenic CO2, we're altering carbonate chemistry, changing the light fields because we're changing the depth of the mixed layer. And so it really is a fundamental shift. It's really difficult to get our head around it. If that wasn't bad enough, we also then have these interactive effects, which we're working on 3 different levels. The physical-chemical level is where we warm the ocean, we change its ability to store this anthropogenic CO2. We also then have effects at the physiological level, and then furthermore at the ecological level. So it's just it's a huge raft of permutations, which makes it difficult to try and make a coherent story out of it, which is really one of the big challenges we have. Because it's not just what's happening globally in the Anthropocene era that we're living in, it's also what's happening regionally and also locally. So anyone in this room who's got a particular critter or bug that they're working on, I imagine that the inventory of the stressors or drivers that they have to look at will differ from locale to locale, from place to place. So again, you know, it requires a sort of almost encyclopedic kind of awareness of what's going with these drivers. And again, it may well be that in your particular neck of the woods that something such as a global driver such as CO2 is not particularly important. It may be something like a point source pollutant. So again, it's important to sort of bear these in mind. And that really causes us a problem because if we don't even know what the inventory of drivers or stressors are for our particular region or particular critter or bugs of interest, then how can we design an experiment? And so this is an example where we've actually tried to come up with some sort of a roadmap. In this case here, we've looked at phytoplankton, we've used the NCAR CSM2 model, we've looked at across 14 biogeochemical provinces, and we've said in the year 2100, can we just blithely assume that we can use the global value for increase in CO2 or changes in nutrients or changes in salinity, or do we have to look more careful in a regional sense? And in brief, what we find is that we do have to look regionally. You can see in the top left-hand corner there the change in pH versus the change in temperature by the year 2100. Because of the melting cryosphere in the Arctic, it's going to be a much more marked influence of a change in pH. I think there was a paper that came out in Nature Climate Change a couple of days ago pointing out a sort of move in this direction. But essentially what we see here is that even across what we think is a fairly homogeneous ocean, regionally we have to come up with, as it were, a sort of a look-up table for each of these regions. I'll put it up very briefly, knowing that this is very late in the day to show something like this. All you need to know is that this is the look-up table you can go to if you're working on open ocean phytoplankton, and in your particular region, for example, the one I've highlighted there is the sub-Antarctic Ocean. You can go there and you can pick out the different anomalies. And what that does give you then is at least a look-up table where you can sort of design experiments with. But of course, that's not the only problem. You know, if we look at you know, a calcareous invertebrate and how it actually will respond to these changes versus a siliceous phytoplankton, and the diatom on the right-hand side, organisms are going to respond differently to an individual driver or stressor, and they're going to respond differently to a combination of those stressors. And so how do we actually ascertain which is the most important thing to actually study? We've got a lookup table, we can design an experiment, but how do we design it further? And so this has really come up with this real dearth of fundamental knowledge. We've got to go back to the 60s and the 70s in many cases to get this very fundamental information, such as thermal reaction norms, or in the right-hand side, a CO2 affinity plot. I know that the study by Hutchinson et al., it took 3 years of painstaking work to come up with a suite of these CO2 affinity curves for a range of unicellular and multicellular diazotrophs in the low latitude ocean. And so it's a case of having to go back in the literature and look up very early work by Dick Epley, John Raven, Larry Brand, and various other sort of pioneers to try and assemble this information. And it's not just in physiology, it's the same with co-limitation, with microevolution, with the whole relationship between diversity and stability. Do we see differential vulnerability across trophic levels? So again, a very challenging sort of problem to deal with. And so this was a nice summary that came out of the sort of decade-long work by the people in ocean acidification by Ulf Ribesil and Jean-Pierre Gattuso. And this is this sort of cube they have. And really, we have to advance knowledge along 3 axes concurrently. We have to move beyond sort of acclamation and look at adaptation and microevolutions on one axis. On another axis, we have moving from species to communities, and then finally up to ecosystems. And for the remainder of the talk, I'll just finish briefly by talking a little bit about how we have to look beyond just a single driver to multiple drivers and take these into consideration. And that's really what we've been focusing on, is trying to tease apart the influence of these drivers. We want to develop a mechanistic understanding. In this case here, we've sort of put IPATH on the left here. We really want to have some sort of molecular underpinning. We want to link that through to the physiology and build some better conceptual frameworks. But one of the challenges we have to do that, if we take phytoplankton, we're looking at concurrent changes in CO2, in pH, in nutrients, in light, in temperature, in trace metals. So, you know, which is the biggest player, and how do we look at these? And one of the slides that I've been using a lot with policy people is this plot here. It's got 9 data points, which are all sea urchins. It's even got error bars if you look at the spines. And what we have here then is a change in pH, one axis, and a change in temperature. We see, if we look down the left-hand side of the figure, that decreasing pH will give us these smaller urchins. But then we see then the opposing effect, that warming temperatures will give us larger urchins. And if we look in the bottom right-hand corner of the graph, we see it's a zero-sum effect. And so this is one of the reasons we've been trying to show this to policy people, because they really don't want to hear beyond ocean acidification. They don't want any more complexity, or there's something we didn't tell you that we're going to tell you now. At least this doesn't give them a sky is falling message that they seem to get all the time. But again, it does illustrate that we have these complex interactions that we have to somehow tease apart. This slide was supplied by Nalgene for a little sort of product placement, because what we have to sort of boil down to in the end then is managers want to know what's the cumulative effect of rebooting the ocean's environmental flashcard. And while they want to know that, then as scientists, we actually want to know then what are the underpinning mechanisms that are driving this matrix of cumulative change. But as you can see, if we just take the example of phytoplankton, where we've got 5 different drivers at least, possibly more in some locales, that's a very large multifactorial experiment that you're going to have to pull off and also interpret. So we've been trying to pick up that challenge and move that along, because really we've got these 2 fundamentally different approaches. On the left-hand side, we have this sort of house of cards we're building, where we might start off with acidification, and then you saw with the example of the sea urchin, that's not the whole story. So that's not really going to be very helpful for managing resources. And so we build the next level carefully, where we might bring in some temperature, we might bring in something else. But ultimately, with that approach, I would argue that you never actually find out what the cumulative environmental change is, because you may not have been manipulating what's the most influential stressor. So again, we get back to this game of permutations. We've taken an opposite approach, where we've gone for a holistic black box approach, where we can throw the kitchen sink, as it were, at these phytoplankton. We can actually get the cumulative effect. And then, of course, we have to try and break into the black box, and we try and do that by teasing apart individual from interactive effects. We try and employ as many diagnoses from omics through to sort of conventional biochemical metrics, at this approach. And so this is a paper from last year where we did this for the first time with a subantarctic phytoplankton, and based on the lookup table that we have, we're going to see major changes by 2100 in temperature, in CO2, in light and nutrients, and also in trace metals. And so we designed this experiment, and what we have to do in the experiment is actually find out, by doing preliminary work, what the dominant driver is. Once we know the dominant driver, we can then collapse this down into 4 treatments as opposed to a very large factorial matrix. And this is how we do it. We ascertain from preliminary work that actually temperature appears to be the dominant, the most influential driver. These are our 4 treatments. So we have a control treatment, we have a future ocean where we just have warming, and that gives us the individual effect of warming. In treatment D at the base of the slide then, we throw the kitchen sink at it, we increase CO2, we warm, we decrease nutrients, we change the light field, and we change the iron. That gives us the cumulative effect of ocean change, which is what we want, one of the targets that we wanted to reach. But also, by comparing it with treatment C, where we alter everything apart from warming, we can then solve for the interactive effects of warming. So this approach with 4 treatments is giving us the individual effect of the dominant driver in the subantarctic for this diatom. It's giving us the interactive effect, and it's giving us the cumulative effect, which are 3 of the things we want to get at. We don't understand everything, and there's only time to show you just as one example. This is some of the proteomics work that Brooke Nunn and Evan Timmins-Schild did. And if we look at this MSDS, you can see this nice. I think it only goes the one way. Oh, the red one, sorry. Yeah. Sorry, if we go back to the slide here, we see this separation of the 4 treatments here, from the control at the bottom, and what we've marked here, then the main either upregulation with the upward arrows or downregulation of these different proteins associated with these different metabolic processes in the cell. The point that I find particularly interesting, but also rather scary, is if we compare treatment D with treatment C, one is the future ocean, the other is the future ocean without warming. We see there's a very big difference between them. And so had we only run with the 4 treatment rather than the full 5 treatment one, we would have got a very different conclusion. So again, illustrating the pitfalls in trying to work our way through this sort of myriad of permutations. As you've seen at this meeting here, we've got a huge number of really impressive tools now, from the molecular work through to nanosim and a whole host of other tools and very sophisticated models. So how can we actually then best use these tools? This is it was supposed to be a Gordian knot, but the art editor at the journal, it looks a bit more like someone's intestines. It's I wouldn't like to put a sword through it, put it that way. But anyway, you can see what we've tried to do here is point out all of these key issues that we have to look at, from trophic cascades through to epigenetics, differential vulnerability. There's a huge number of things that we need to tackle, and we probably have the tools to do it. However, there are problems. This is again from the ocean acidification literature. This is a sort of synthesis that Hop et al. Did on Emiliana Huxleyi. If you look at just the little vignettes, that they basically give you the trends. In some cases there are no changes, in some cases there are non-linear changes, in other cases there are increases, in some there are decreases. And having been involved in Working Group 2 chapter on ocean systems in AR5 of IPCC, trying to make sense of this and trying to write something sensible and coherent really is a challenge. And so this is just for ocean acidification. We haven't looked at any other drivers in addition to it. And so this puts us in this position where there's almost a paradox, such that there are graduate students out there at the minute who are running microevolution experiments over 600 generations, and they're done in such a unique way that n-1 equals 0. There are, if you like, a scientific solipsism in terms of an experiment. And so we really have to think carefully. These experiments are important, but we need, if we're going to form a coherent picture, we need some way that we can actually try and work around it, some way we can cross-reference it. If you look at things like the GeoTraces Global Survey, chemists from around the globe have standards that they can cross-compare data sets and build a picture. And so the challenge or the question we have to ask is there some way that we can also do the same to try and share the load and share the heavy lifting? And so I'll just finish having talked the talk to ask the question, can we actually then walk the walk? And there are a number of different groups out there starting to move stuff along. ECO was an NSF sort of catalytic workshop that brought experimental evolutionary biologists together with oceanographers. It was a very interesting meeting for 3 days, but different lexicons and other challenges. Programs like IMBER have been moving into this work with their Future Oceans program. The Gordon Conference has been going now for 2 conferences now that was initiated by David Hutchinson and myself. So we are starting to build a little bit of a community, but we have to start to think about how we can cross-reference our research. We had for ocean acidification the best practice guide. Some people thought it was useful, other people thought it was a bit bossy and prescriptive. And so we're asking questions now to say, can we have biological reference organisms that might be centered around organisms that have had their genome published? There are challenges with that also. Or another example is this pilot study where we tried to do a community-wide study across 7 labs using common protocols. And so recently we've been able to have this working group funded through SCORE, and you can see that quite a few people there from ocean acidification research who we're learning from their advances, but also some of their mistakes as part of their sort of learning curve. And last year we also had a workshop as part of the Oceans in a High CO2 World meeting where we asked a lot of questions of some of the new early career researchers who are coming through to find out about modes of learning. And so to try and to finish this short presentation on a high note, we've now got this new plan for this hopefully fairly nimble web-based best practice guide for multiple drivers. So rather than having a volume, 6 or 7 volumes on nutrients, temperature, oxygen, and other drivers, we're actually going to try and come up with something to help guide people through and help them learn. So it has 3 tiers, a web-based resort with flowcharts where you can basically work out what might be the best experiment or what might be the stressor inventory for your critter or your locale to help people sort of steer a path through it. And then we just raised sufficient funding then to set up a web-based self-learning in a virtual laboratory where you actually go and design your experiments and you make your mistakes in silico rather than spending a year doing something and then realizing it wasn't really practical. So we're going to spin that up. And then finally the 3rd tier will be, with some influence from ASLO then, a series of webinars designed by a series of experts. And it means people can come in, be it a newcomer or someone experienced in the field, and they can customize how they do their learning, and hopefully that'll move us away from this other sort of book approach which people thought was a little bit bossy. So anyway, I'll leave it there. Thank you very much again for patiently waiting and staying to the end of this. It's really been a thrill to accept this award, particularly the Hutchinson Award. So thank you.